Okay, let's talk about genome interactions because this is pretty cool. So, our genome is pretty much like our complete genetic makeup. The human genome was mapped and was completed in 2000s, the early 2000s, which is a pretty astonishing accomplishment since the human gene has about 25,000 genes in it. The great part about all of this is that it shows how diverse life really is. And it's really a universal code made up of the same nitrogen bases, such as adenine, thymine, guanine, and cytosine for DNA, and for RNA, guanine, cytosine, uracil, and adenine. So it's pretty crazy that we have this universal code that everybody can do that pretty much codes for the same amino acids. If you think about that, if we have the codon, again, codons are found on mRNA, GUC, that also codes for valine in bacteria, eagles, and humans and plants. Like, that's crazy. Even though some organisms don't need 20 amino acids like us, and they have many, many less. Others have a little bit more, but the whole deal is we're made up of this universal code. That scary part can be interchanged, and we do interchange it. The genome size is relative to the amount of DNA that they have. So the more DNA you have, well, there we go. So let's look at a genome for the proteins of the different coronaviruses. Right now, we are dealing with coronavirus that causes our COVID-19. And so those are a little bit different. But if we look here, this is kind of what we call an electrophoresis. And we can start to see these lines show similarities. So they should match. So like these two here match for so for our SARS, CoV, these guys match right here. And then we see the same, and then you can see your differences starting to occur right here. And that gives us a genetic variation. So we're very similar, and here's our next SARS-CoV, and we see similarity, and then right here in the same little section, that's where we're having our differences. And so when geneticists run these, we can see if under an electrophoresis that these all match up and they're exactly the same, then they would be the exact same thing. But because we have some differences down here, we can tell, okay, everything from pretty much here back is exactly the same. So they have to be related to one another. And they are. If we start looking at this one, this is the MERS-CoV, so it's a little bit different. You can see that the S is much longer, and then our first E is shorter, and then we go, so there's a little bit of a difference there. So the genetic diversity of these can just be just a very small amount. The problem with it being just a small amount is that our immune system doesn't necessarily have an antibody for that specific protein. Therefore, we don't have immunity to it. It's not uncommon for things like a coronavirus, which for the most part causes cold-like symptoms, or influenza to change rapidly. And we have vaccines. So the influenza vaccine that you get every year, the flu shot, is based on calculations of what they think is going to be the mutation that occurs for the next week. But let's look at the coronavirus and human origin. So this is where we look at, say, okay, what was the host that gave us our COVID-19? We don't know. Well, actually, we do. We think it's a pangolin. So here's a natural host. These are bats here, and then mice for some. So bats are kind of like little kids. And I say that in the nicest, warmest way possible. So a bat's immune system is like a little kid's immune system. Little kids really don't have a lot of immunity except for what their mom gives them. And we are shooting little kids up with vaccines so that they're gaining antibodies, and they're just like, they have a system overload. And so to deal with that, they were like, okay, you're okay. And they kind of just take care of it and move on. And that's what a bat's immune system does too. And it doesn't like stress out about that one antibody, it just kind of like, okay, we got antibodies, let's go. Let's go to the next one. Okay, process of elimination, we got to get through all these. Where in a human who is older, old people, I get it, I'm one of them. Our immune system goes, no, we will take care of this until it's completely eradicated from the body. And we focus on something, our immune system focuses on that so much that it can actually cause autoimmune disorders. And so that becomes a little bit of an issue. So our immune system is kind of working overtime for that one pathogen, and then we become sick with things like that sneak in on the back door. So that can become kind of a iffy situation. But bats don't have that problem. They're like little kids for life. Oh, let's deal with you, and then let's deal with you, and then they just keep moving on. So if we kind of look at some of these intermediate hosts, SARS, kind of like a cat-like ferret fox deal. We have pigs, MERS. We have our camels, cows, and our llamas. And so we're trying to figure out what that intermediate host is that passes on these coronaviruses to us. We know that they are a zoological one, so they're coming from something that we're eating. By the way, you don't really get these if you process your food correctly. So as long as you have a clean kitchen and you cook your food up to temperature, you don't have to worry about some transmutations. So let's talk about DNA versus genes. So first off, let's talk about bacteria. This is a bacterium. Notice there's DNA and then there's plasmids. We like to mess with the plasmids a lot because they're easy. They're circular. They're usually easily manipulated. We can transpose pieces. They're super fun for us to do. A lot of times they have some extra pieces of plasmid. And so one of the cool things that we can do with this is we can actually alter the genetic structure. So here we have a bacteria, DNA, plasmids. What if we did something a little different? Notice cell replication, DNA, plasmids. Here's something a little bit different though. We're integrating the plasmid into the DNA. We've now integrated that. So now we've changed the DNA sequence. This is really easy to do in bacteria because their DNA is naked and sitting out there for all of us, and we can integrate the plasmid. Oh wait, they do that to themselves already. So for us, we just learned from them how to do this, and so now we can do it to a lot of things. We like to do it a lot to plants. They don't speak up a lot. It's kind of nice. However, in eukaryotes, there's something a little bit different. We talked about this a little earlier. We have the coding sections and the non-coding sections, the exons and the introns. And so the function of most of the non-coding DNA is really unclear. We know it's like a space holder, but we're not really sure why it's a space holder. We also know that sometimes with mutations, it will take the coding DNA and add a little piece of the non-coding DNA and make that now a coding piece, which can cause some good mutations and some bad mutations. It all kind of depends. And so what we do know is that the genomes that we have, they will code for proteins. And we know that is the central dogma, DNA to RNA to proteins. Now, our organelles have a function that separates them from everything else within that cell. So like the mitochondria is producing ATP while the lysosomes are digesting waste products and nutrients so that we can have a clean internal stuff. So we have all these specific functions, and so they have to make these specific proteins to get there. And now that we start thinking about this, how do we have all these organelles that do all these specific jobs? They have a theory called the endosymbiotic theory. So let's break down the word endosymbiotic before we start talking about all of these questions that I have about the endosymbiotic theory. The endosymbiotic, endo, inside, symbiotic relationship. So there's a mutualistic relationship occurring, and it's a theory. So it's not an educated guess. This has been proven scientifically over and over and over and over and over again, but it can't become a law because, dun, 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 laws happen everywhere in the universe. And I don't know about you, but they haven't proven that it happens everywhere in the universe. And I know Star Wars fans everywhere are screaming, but it's true. We haven't found life on another planet that we can actually go back and study. So it can only be a theory until we find other life. There we are. So what is the endosymbiotic theory? Why is it dealing with mitochondria and chloroplasts? Who is the person that came up with this? Why is it important? How does it apply to genome interactions? And why do some people think, well, it's not really valid? And does it really relate to symbiosis? Well, here we go. The endosymbiotic theory. When we start talking about the endosymbiotic theory, what is it? Well, we pretty much say I was engulfed by a giant cell. Well, that's pretty much true. So here's what we think happened well before life with us. So about 1 billion plus years ago, we have a single-celled organism engulfing and usually digesting another cell. However, it didn't digest it. It kind of hung out there. We believe that the two cells that are responsible for this are chloroplasts and mitochondria. And the reason for that is because you have a different DNA sequence within your chloroplast and your mitochondria that is not found within your nucleus. In fact, everybody gets mitochondrial DNA from your mother. You don't have any paternal, it's just maternal. So we know that those two operate a little bit differently and separately. We also know that these guys, the chloroplast and the mitochondria, are able to produce energy. And here's the fun fact. Mitochondria came first. How do we know that? Well, plant cells have mitochondria, where most animal cells, if you're not photosynthetic, you don't have chloroplasts, but all plant cells pretty much have mitochondria in there. So we know that they came that way. So chloroplasts and mitochondria evidence. One, they're membranes. Some of them have a double membrane, which means it's a little bit different. However, not all organelles have a double membrane. It's kind of specific. They're susceptible to antibiotics, which is different. Those two specific organelles are susceptible to antibiotics. They reproduce in a fission-like process. That's kind of crazy if you think about it. The DNA has its own specific markers separate from the DNA of the nucleus, and the ribosomes are about the same size as a prokaryotic ribosome. That's kind of crazy if you think about it. But where did all of this come from? It came from a scientist called Lynn Margulies. So Margulies developed this in 1973, and from that, she was awarded the Presidential Award. However, she did pass away in 2011, and so another great woman scientist has passed. But she did leave us with the endosymbiotic theory. It is, however, controversial because it kind of shows the process of evolution. And we would say, like, the endosymbiotic theory doesn't happen today, and we can't prove it, but we're actually seeing it happen today. We can make it happen in labs, which is pretty cool, though. You can think about that. So let's go beyond. And let's talk about mobile genetic elements, or MGEs. These are units of DNA that can sometimes be transposed or removed. Transposon is when a genetic sequence is randomly moved to a new place within the genome, and it can function within that place within the genome. Some bacteria are able to transposon all the time between plasmids and chromosomes. Viruses are small and non-living, but yet they can complete transposon, which is kind of crazy if you think about the interaction of the variety of genetic material that we have, and we can actually see that that happens. So here's an example of that. So we have all of these interacting together, and now 4 became 2. So we can actually genetically engineer organisms that are completely new within a laboratory, especially if they're prokaryotic organisms. And so here's what we can see. We have this normal functioning gene that's producing the APC product, and then we have a transposon insertion. So we're inserting a line of DNA, and now all of a sudden, because we have this AP, we've now separated it with a line and then have a C separate. It will no longer produce that product anymore. And so this is something that we've done to things like papaya so that they can have an immune system, because plants don't have an immune system. And if we didn't do something like transposon, we wouldn't have any papaya. Here's what viruses do. So this is our viral genome in blue up here. And what they do is they come in contact with in this case, bacteria genome. And they're actually going to have a transposon. And by the time that we're all said and done with this, when the bacteria actually replicate, it's now got viral genome stuck inside of its genome. The great part about this is now the virus can program the bacteria to just make more viruses for it. Assembles all the protein and everything. It's kind of sad. And we'll do it until it's so full that it explodes virus everywhere. So these mobile genetic elements are extremely helpful for us to understand. And they're also really cool because then they allow for us to make genes so that they can maybe do functions that we didn't know that they could do before. We're using this for things like gene therapy so that we can replace proteins that our body can't make for some reason, and now they can actually start to make them. The other kind of scary part about this is antibiotic resistance. Like mutations, transposons have helpful and harmful effects. And so one of those would be antibiotic resistance. And for us, that becomes really important. Note, when we're talking about antibiotics, we're talking about bacteria, not viruses. So let's look at this. How does antibiotic resistance work? Well, we have lots of lots of cells within our body. You take an antibiotic. The antibiotics will kill the bacteria, causing the illness to hopefully go away. However, see the blue one? Those become resistant to bacteria. Part of the ways it can do this is because every time it's exposed to that bacteria, it actually starts to gain some resistance. Every time you kind of take ibuprofen or Tylenol, your body gains a tolerance to it, and then you have to take more and more and more if you chronically use something like that. So this happens kind of the same way. So when you're given antibiotics, you should stop and take it for the whole time, okay? Give me 1 second. And when you take that, then you don't leave any of those antibiotics there. Even if you're feeling better, you should take all of your antibiotics. The problem is that we don't always do that. And then what happens is the next time you get sick, it's not sick with the bacteria that were killed by the antibiotic, but you're sick with the bacteria that now have a resistance to it. This means a couple things. 1, we have to find new antibiotics. Or 2, we have to up the dose of the current antibiotics. And some bacteria can have their drug resistance passed on to other bacteria, and that's the transposon that we're talking about that. And so when we start talking about hospitals, you'll notice that one of the diseases that we're really worried about is MRSA. MRSA is caused by Staphylococcus aureus. However, here's the deal with Staphylococcus aureus. It's all over you, on the outside of your body. It's actually a healthy bacteria that lives on the outside of your body. However, it should not be on the inside. Remember we talked about Escherichia coli and bacteria within your colon? Those things are really great. Gut bacteria is super healthy for your gut. However, anywhere else in your body, it can go septic. MRSA is the same way. Staphylococcus aureus is all over you. So it's pretty common to find when we start collecting bacteria samples. However, Staphylococcus aureus inside of you can lead to things like MRSA, which is antibiotic resistant because if you think about it, you wash your hands, you take showers, you do all this stuff, but you still have this bacteria on you. It's because it's now gained a resistance to it. That's one of the reasons it's really hard for this. And that is the last section of notes.